Good afternoon, everyone. My name is Maria, and today I will discuss a recent Dalton transition paper written by James and his co-workers, which focuses on developing their green and synthetic roots of ruthenium-2 pyribaldyl complexes and evaluating whether this impacts their photophysical and biological behavior. These works are interface of coordination chemistry, photophysics, and chemical biology, and is particularly relevant because these complexes are widely used as a DNA probes, yet are still synthesized with environmentally problematic solvents. To understand why this chemistry is interesting, it helps us to first look at how these complexes behave electronically. If we look at their energy diagram on the left, they show what happens when the BPZ-based ruthenium-2 complexes absorb light in a non-aqueous solution and environment. Excitation populates an MLCT state that can relax by emitting light, given a strong luminescence. But if we look at the diagram below, representing water, the situation changes. Here, the interactions which solvent introduce the additional non-radiative pathway, so the excited state is rapidly quenched. This difference between the environments is subtle, but is turned out to be incredibly powerful because it means emission is controlled by whether the complex is. DNA provides the exact kind of environment. If we can have a look at the structure on the left, we can see how planar metal complex intersect between two DNA base pairs through intercalation. By sitting inside the DNA stack, the complex is physically shielded from the water, which restores the MLCT emission. On the right of the table, we highlight the difference between well-matched and mismatched DNA sequences. Mismatched destroys the DNA helix and creates locally destabilized region. These sites are often more accessible for bulky interactions, making them particularly attractive targets. This is why ruthenium-2 light switch effect complexes are not just fluorescence molecules. They can report on subtle structural features of DNA that are biologically relevant, including errors associated with disease. As we can see here, their scheme shows us how to synthesize ruthenium-2 pyribaldyl complexes. The most important part here is to synthesize the intermediate complex using the solvent, and the solvent is being used as a DMF. The important thing here is DMF is not just a solvent. It is reducing ruthenium-3 to ruthenium-2. But, however, we do know that a DMF is very highly toxic solvent and very bad for environment. So scientists are particularly was focusing what solvent we can use to substitute DMF, but be more environmentally friendly. Cyrene was brought as a potential solvent. Cyrene behaves structurally and chemically like dipolar aprotic solvent, which means it can dissolve inorganic salts and pyribaldyl ligand much like a DMF. At the same time, it's biodiverse and biodegradable, originating from cellulose waste rather than fossil resources. The key question here is not if Cyrene is greener, but whether it can survive the harsh conditions required for ruthenium-2 coordination chemistry without binding to metal or decomposing. This is what makes this substitution chemically interesting rather than cosmetic. So, as we can clearly see on the table on the left, the authors approached that optimization. Early attempts with high temperature led to solvent degradation, which immediately tells us that the Cyrene cannot simply be treated as a drop-in replacement. By reducing the temperature and time reaction, they were clearly be able to form ruthenium BP2Cl2 complex, which with yields comparable, or in some cases higher than obtained in DMF. What's more interesting for me here is the striking result with a microwave, where the reaction was completed in minutes rather than hours. This demonstrates that Cyrene is not only compatible with the chemistry, but also is more energy efficient synthetic techniques. Well, and here authors are directly comparing green synthesized ruthenium BP32 with a commercial reference. The NMR structure shows identical chemical shapes and peak patterns, indicating the same coordination environment. Similarly, if we have a look at HPLC traits overlap almost perfectly, demonstrating compatible purity and retention behavior. Together, these features provide a strong experimental evidence that replacing DMF with serine does not alter chemical identity of the final complex. So the plot here shows what happens when methylated DPPZ complexes interact with the DNA. So authors taken 3 different complexes, 2 of which was methylated DPPZ, and 1 of them was DPPZ, which is parent complex. And comparing this parent complex to emission intensity, increasing dramatically indicated as much stronger light switch effect response. The lifetime data add another layer to the story. Emission now persists in microsecond timescale, which is unusual long for ruthenium-2 complexes. This combination of high intensity and long lifetime is particularly valuable for bioimaging, where background fluorescence is a major limitation. To move beyond qualitative trends, the authors then quantify how strongly these complexes bind to the DNA. So as we can see here on the figure, the binding curves quantify DNA affinity using the Fred-based assay. The difference between the curves clearly shows that the dimethylation of the DPPZ ligand increases binding strength. What's important here that this closes the loop of the story. A green synthetic route produces chemically identical complexes which retain and in some cases enhance their DNA binding and photophysical performances. Their sustainability improvements therefore comes without sacrificing function. So if we look at these fluorescence microwave images, we can see that the green synthesized complexes will behave exactly as functional chemistry probes in cells, co-localizing with the plasma membrane and staining in their nucleus. This is visual proof of the green synthesis does not compromise imaging performance. What this really signals to the field is something bigger. A solvent can be substituted and compatible with the metal reduction and emulsivity chemistry, with coordination chemists have historically assumed to be manually exclusive. This paper is not just synthesis upgrade, it expands what we consider chemically possible under the regulation's safe conditions. So looking at the ligand now, we can clearly start see that Fred result did not behave the way they expected. Methyl group were added at a position 10 and 11. The energy levels of the complex shifted more than traditional modes predicted. This tells us and them that even small structural changes on the ligand can have a surprisingly large effect on how complexes absorb the emission light. Further studies using the computational chemistry tools like DFT could help measure exactly how the energy changes and also simulate how the complex fits into a normal DNA versus DNA that contains mismatches or distortions. If we zoom in into methyl positions, highlighted here, another promising idea would be methylation at positions 10 and 13 instead. These sites may protect the ligand from water quenching even better, while binding less strongly to perfectly matched DNA. That could make the complex more selective for mismatches, shifting it from being a general DNA binder to more diagnostic, structural sensitive probe. Thank you very much for your attention.